In 1951, a young mother named Henrietta Lacks died of cervical cancer in a Baltimore hospital. She was 31 years old. Her body was buried in an unmarked grave in Virginia, but part of Henrietta didn't die that day. Part of her is still alive, still growing, still reproducing. 70 years later, Henrietta Lacks' cells have been to space, helped develop the polio vaccine, contributed to COVID research, and exist in laboratories on every continent. Scientists have grown so many of her cells that if you could weigh them all, they would weigh more than 100 Empire State Buildings. More mass than Henrietta's original body by a factor of thousands. This is the story of the woman who became immortal without knowing it, whose cells changed medicine forever, and whose family didn't find out until 20 years after her death. This is the story of HeLa cells, the most important biological material in medical history, and the ethical questions that still haunt us today. Chapter 1. The Woman Behind the Cells, Henrietta Lacks' Life. Henrietta Pleasant was born on August 1, 1920 in Roanoke, Virginia. Her mother died when Henrietta was 4 years old, and she was sent to live with her grandfather in a log cabin that had once been a slave quarters on a tobacco plantation. The cabin had no running water, no electricity. Life was hard for black families in rural Virginia in the 1920s and 30s. Segregation was law. Opportunity was scarce, but Henrietta was vibrant, full of life. Her cousins remembered her as someone who loved to dance, who had a beautiful smile, who took care of everyone around her. At 14, she gave birth to her first child. At 20, she married David Lacks, her cousin, and they moved to Baltimore, Maryland, seeking work in the steel mills and shipyards. It was the Great Migration, when millions of black Americans left the rural South for industrial jobs in the North. Henrietta and David had 5 children together. She worked hard, took care of her family, went to church. By all accounts, she was a devoted mother, a loving wife, an ordinary woman living an ordinary life in extraordinary times. America was transforming. World War II had just ended. The baby boom was beginning. Medical science was making unprecedented advances. And Henrietta was about to become the most important person in medical history, though she would never know it. In January 1951, Henrietta noticed something wrong. She was bleeding when she shouldn't be. She felt a knot inside her. She went to Johns Hopkins Hospital, one of the few hospitals in Baltimore that treated black patients. The gynecology clinic was segregated, black patients in one waiting room, white patients in another. But Johns Hopkins had a reputation for quality care, and it was free for those who couldn't pay. The doctors examined Henrietta and found a tumor on her cervix. They took a biopsy, cutting a small sample of tissue to send to the laboratory for analysis. The results came back quickly. It was cancer, aggressive, fast-growing cervical cancer. Henrietta began treatment immediately. The standard treatment in 1951 was radium, a radioactive element that doctors would insert directly into the tumor site, hoping to kill the cancer cells with radiation. It was brutal treatment, painful, dangerous, but it was the best medicine had to offer. During one of her treatments, the surgeon took another sample of tissue from Henrietta's cervix, one sample from the tumor, one sample from healthy tissue nearby. These samples were sent to the laboratory of Dr. George Gay, a researcher who had been trying for years to grow human cells and culture outside the body. This was the holy grail of medical research at the time. If scientists could grow human cells in the laboratory, they could test drugs without using human subjects. They could study diseases and control conditions. They could revolutionize medicine. But every attempt had failed. Human cells, when removed from the body, would survive for a few days, maybe a few weeks, and then they would die. Dr. Gay had tried thousands of samples from thousands of patients. Nothing worked. The cells always died, until Henrietta's cells. When Dr. Guy's assistant, Mary Kubitschek, prepared Henrietta's cells in a culture dish, adding nutrients and storing them in an incubator, something extraordinary happened. The cells didn't just survive, they thrived. They doubled overnight, then doubled again and again. By the time Mary checked on them a few days later, the cells had taken over the entire dish. They were growing faster than any human cells ever observed. They were, in a word that would come to define them, immortal. Dr. Gay could hardly believe it. He had finally succeeded in creating an immortal human cell line. He named them HeLa, taking the first two letters of Henrietta's first and last names. He began sharing them with researchers around the world, free of charge. Anyone who wanted to study human cells could request a sample of HeLa. Meanwhile, Henrietta's cancer was spreading. Despite the radium treatment, despite everything the doctors tried, the tumor was growing, metastasizing throughout her body. She was in terrible pain. The cancer was eating away at her from the inside. On October 4th, 1951, 8 months after her initial diagnosis, Henrietta Lacks died. She was 31 years old. She left behind a husband and 5 children, the youngest just an infant. Her family, devastated by grief, buried her in an unmarked grave in Clover, Virginia, near the log cabin where she'd grown up. And that should have been the end of Henrietta's story. A young woman, dead too soon from a terrible disease, a tragedy, but a common one. Cervical cancer killed thousands of women every year in 1951. But Henrietta's story didn't end. In fact, it was just beginning. Because while Henrietta's body lay in the ground, her cells were alive in laboratories around the world, reproducing, spreading, changing medicine forever, and her family had no idea. Chapter 2. The Miracle Cells. How HeLa Changed Everything. Dr. George Gay understood immediately that HeLa cells were revolutionary. Within months of Henrietta's death, he was shipping samples to researchers across the country. The response was overwhelming. Scientists who had struggled for years to grow human cells in culture suddenly had access to an unlimited supply. HeLa cells became the workhorse of medical research. They were used to test the effects of radiation, toxins, drugs. They helped researchers understand how cells divided, how cancer spread, how viruses infected human tissue. They were cheap to maintain, easy to grow, and apparently indestructible. But the first major breakthrough came in 1952 when Dr. Jonas Salk was developing a vaccine for polio. Polio was one of the most feared diseases in America. Every summer, parents lived in terror that their children would be infected. The disease caused paralysis, left children in iron lungs, killed thousands every year. Salk had developed a promising vaccine, but he needed to test it. He needed to grow large quantities of polio virus, and to do that, he needed human cells. Traditional methods required kidneys from monkeys, which were expensive, limited, and ethically problematic. HeLa cells solved the problem. Salk could grow unlimited quantities of HeLa cells, infect them with polio virus, and use the resulting virus to produce vaccine. The cells grew so fast, so reliably, that mass production became possible. In 1954, Salk's polio vaccine was tested on nearly 2 million children in the largest medical trial in history. It worked. Polio rates plummeted. Within a few years, a disease that had paralyzed thousands of children annually was nearly eliminated in the United States. HeLa cells had helped save an entire generation of children, but the public didn't know the cells came from a black woman named Henrietta Lacks. Most people didn't even know what HeLa stood for. The cells were just referred to as HeLa, a scientific designation with no connection to a human being who had lived and died. The success of the polio vaccine made HeLa cells even more valuable. Demand skyrocketed. Researchers wanted samples for every kind of study imaginable, and supplying that demand became a business. A scientist named George Gay had been giving HeLa cells away for free, believing that science should be shared openly. But others saw commercial potential. Companies began culturing HeLa cells in massive quantities and selling them to laboratories. The cells were shipped around the world, generating millions of dollars in revenue. None of that money went to Henrietta's family. In the 1960s, HeLa cells went to space. NASA wanted to understand how human cells behaved in zero gravity, whether space travel would be safe for astronauts. They sent HeLa cells on multiple missions, studying how they divided and grew without Earth's gravity. The cells thrived in space just as they did on Earth. Henrietta's cells had literally left the planet. In the 1970s, HeLa cells helped develop treatments for leukemia, influenza, hemophilia, Parkinson's disease. They were used to understand how cells responded to chemotherapy, helping optimize cancer treatments that saved countless lives. In the 1980s, when the HIV/AIDS epidemic emerged, HeLa cells were crucial in understanding how the virus infected human cells, leading to the development of treatments that turned AIDS from a death sentence into a manageable condition. In 2020, when COVID-19 became a global pandemic, researchers used HeLa cells to study the virus, test treatments, develop vaccines. Henrietta's cells, nearly 70 years after her death, helped save lives during a once-in-a-century pandemic. By conservative estimates, HeLa cells have contributed to more than 75,000 scientific studies. They've been used in research that led to 11 Nobel Prizes. They've helped develop countless drugs and treatments that have saved millions of lives worldwide. Scientists have grown approximately 50 million metric tons of HeLa cells over the past 70 years. That's more than the weight of 100 Empire State Buildings. It's thousands of times more mass than Henrietta's original body. If you laid out all the HeLa cells ever grown end to end, they would wrap around the Earth 3 times, or reach to the moon and back. The sheer quantity of living Henrietta Lacks tissue that exists today is staggering. And yet, for 20 years after Henrietta died, her family didn't know that her cells were alive. They didn't know they'd gone to space. They didn't know they'd helped develop the polio vaccine. They didn't know her cells existed in laboratories on every continent. They didn't know because nobody told them. Chapter 3. The Family Discovers the Truth. In 1973, a scientist contacted Henrietta's husband, David Lacks. The scientist needed blood samples from Henrietta's children for research. He wanted to study their DNA to better understand HeLa cells, to create genetic markers that would help identify the cell line in laboratory settings. David was confused. He didn't understand what the scientist was talking about. What cells? His wife had been dead for more than 20 years. The scientist explained casually that Henrietta's cells were still alive, that they were being used in laboratories around the world, that they were the most important cells in medical research. David was shocked. His children were shocked. They had no idea. Nobody from Johns Hopkins had ever contacted them. Nobody had ever asked permission. Nobody had ever explained what had happened. The scientist wanted blood samples, but he didn't explain why clearly. Henrietta's children, with limited education and no understanding of medical research, thought they were being tested to see if they had the same cancer their mother had died from. They were terrified. Deborah Lacks, Henrietta's youngest daughter, was especially upset. She was an infant when her mother died. She had no memories of Henrietta. And now she was learning that part of her mother was still alive, still existing somehow in laboratories, and nobody had thought to tell the family. Deborah started asking questions. She contacted Johns Hopkins. She tried to find scientists who were working with HeLa cells. She wanted to understand what had happened to her mother, why her cells were different, what it all meant. The answers she got were confusing, technical, often condescending. Scientists would tell her about cell biology, about immortalization, about research protocols. But what Deborah wanted to know was simpler and more profound. Was part of her mother still alive? Could her mother feel anything? Where were the cells? Could she see them? These weren't questions that scientists were equipped to answer because they weren't really scientific questions. They were human questions about identity, about death, about what it means to exist. Over the next 30 years, Deborah Lacks devoted herself to learning about her mother and the cells. She collected scientific papers she couldn't understand, attended conferences where researchers discussed HeLa without mentioning Henrietta, tried to make sense of the strange legacy her mother had left behind. She learned that HeLa cells had been used in ways that would have troubled Henrietta. They'd been used in weapons research, testing biological agents. They'd been used in cosmetics testing. They'd been sent to laboratories in countries around the world without any oversight or ethical review. She learned that companies were making millions of dollars selling HeLa cells, while her family couldn't afford health insurance. Her mother's cells had contributed to countless medical breakthroughs, but the Lacks family struggled to pay for their own medical care. She learned that her mother's cells had been shared, studied, and commodified without any consent, without any compensation, without even the basic courtesy of notifying the family. And perhaps most disturbing, Deborah learned that her mother's medical records, which should have been private, had been published in scientific papers. Henrietta's name, her medical history, details about her disease and her life were available to anyone who cared to look. The concept of medical privacy, informed consent, patient rights, these things did exist in 1951, but they weren't consistently applied, especially not to Black patients, especially not to poor patients. Henrietta's cells were taken without her knowledge during a medical treatment. This was legal at the time, technically, but that legality doesn't make it ethical. In 2010, a writer named Rebecca Skloot published a book called The Immortal Life of Henrietta Lacks. Skloot had spent a decade researching Henrietta's life, working with Deborah Lacks to tell the full story. The book became a massive bestseller, finally bringing Henrietta's story to the public. Suddenly, millions of people knew about Henrietta Lacks. They knew her name, her face, her story. She was no longer just HeLa, an abstract cell line. She was a real person, a woman who had lived and died, who had been taken from without consent, whose family had been ignored and excluded from the enormous scientific and commercial enterprise built on her cells. The book sparked a necessary conversation about medical ethics, informed consent, the exploitation of vulnerable populations in medical research. It forced the scientific community to reckon with its history, to acknowledge that much of modern medicine was built on practices that we now recognize as deeply unethical. In 2013, the National Institutes of Health reached an agreement with the Lacks family. Going forward, the family would have some say in how Henrietta's genetic information was shared. Scientists who wanted to use HeLa cells for research involving Henrietta's genome would have to apply for permission from a review board that included Lacks family members. It was a small acknowledgement, decades too late, but it was something. Deborah Lacks died in 2009, just before the book about her mother was published. She spent her entire life trying to understand what happened to Henrietta, trying to reconcile the fact that her mother was both dead and somehow still alive. She never got all the answers she wanted. The questions were too big, too strange, too tangled up in science and ethics and grief. but she made sure the world knew her mother's name. Chapter 4. The Science of Immortality. Why HeLa cells won't die. So what made Henrietta's cells different? Why did they survive and multiply when every other human cell line had failed? The answer lies in the cancer that killed Henrietta, and in a remarkable property that cancer cells sometimes develop. Normal human cells have a built-in limit on how many times they can divide. This is called the Hayflick limit, discovered in the 1960s. Human cells can typically divide about 40 to 60 times before they stop dividing and eventually die. This is controlled by structures at the ends of chromosomes called telomeres. Think of telomeres like the plastic tips on shoelaces. Every time a cell divides, the telomeres get a little shorter. Eventually, they're too short and the cell can't divide anymore. It enters a state called senescence, essentially retiring from active duty. This is one of the mechanisms that protects us from cancer, actually. It limits how many times damaged cells can reproduce. But cancer cells often find ways around this limit. Henrietta's cancer cells had activated an enzyme called telomerase, which rebuilds telomeres, keeping them from getting shorter. With telomerase active, the cells could divide indefinitely. They had escaped the Hayflick limit. They had become, in the technical sense, immortal. But that alone doesn't explain why HeLa cells were so much more robust than other cancer cell lines that researchers had tried to culture. There's something else unusual about HeLa cells, something that wasn't discovered until decades after Henrietta died. In 2013, scientists sequenced the HeLa genome. What they found was startling. HeLa cells don't have the same number of chromosomes as normal human cells. Normal humans have 46 chromosomes, 23 pairs. HeLa cells have 76 to 80 chromosomes, and they're wildly rearranged. Pieces of different chromosomes have broken off and fused to other chromosomes. The genome is a chaotic mess compared to normal human DNA. This chromosomal chaos is actually part of why HeLa cells are so resilient. The extra genetic material, the duplicated genes, the rearrangements, they all give HeLa cells redundancy and flexibility. If one copy of an essential gene is damaged, there are backup copies. If environmental conditions change, the genetic diversity allows some cells to adapt and survive. HeLa cells are also infected with HPV, human papillomavirus, the virus that caused Henrietta's cervical cancer. An HPV integrates its DNA into the host cell's genome, and it produces proteins that disable the cell's normal safety mechanisms. Specifically, HPV proteins block p53 and RB, two critical tumor suppressor genes that normally prevent uncontrolled cell division. With these safety mechanisms disabled, with telomerase active, with a chaotic but robust genome, HeLa cells became a kind of supercell. They could survive conditions that would kill normal cells. They could divide indefinitely without triggering the safeguards that normally stop runaway cell growth. Scientists have grown HeLa cells in every conceivable condition, frozen in liquid nitrogen for decades, then thawed and grown again, exposed to radiation, to toxins, to extreme temperatures, sent into space, deprived of oxygen, subjected to drugs designed to kill cancer cells. HeLa cells survive all of it. Not every cell survives, but enough do that the population recovers and continues growing. They're remarkably, almost frighteningly, hard to kill. There's another peculiarity about HeLa cells that has caused problems in scientific research. They're aggressive colonizers. If even a tiny number of HeLa cells contaminate another cell culture, they'll quickly take over, outcompeting the other cells. They grow faster, survive better, dominate the environment. In the 1960s and 70s, scientists discovered that many cell lines they thought were from different tissues Different donors, even different species, were actually HeLa cells in disguise. HeLa had contaminated laboratories around the world, spreading through shared equipment, through aerosols, through careless technique. Studies that researchers thought they were conducting on liver cells or kidney cells or mouse cells were actually being conducted on HeLa cells. Decades of research had to be questioned. Papers had to be retracted. It was a massive scientific crisis, one that the research community was slow to acknowledge and address. Even today, with better laboratory practices and authentication methods, HeLa contamination remains a problem. The cells are so resilient, so fast-growing, so opportunistic, that they continue to show up in places they shouldn't be. In a way, HeLa cells behave almost like an invasive species. They're optimized for survival and reproduction in the artificial environment of a laboratory. They outcompete other cells. They spread beyond their intended boundaries. They're nearly impossible to eliminate once they're established. Scientists studying HeLa cells have discovered other remarkable properties. The cells can adapt to different growth media, different nutrients. Colonies of HeLa cells grown in different laboratories, under different conditions for decades, have evolved differently, developing distinct characteristics. They're evolving in real time, adapting to their environments, becoming specialized for the specific conditions they're grown in. In a technical, biological sense, you could argue that HeLa cells are no longer quite human. They have human DNA, but so much of it has been rearranged, duplicated, deleted. They have HPV DNA integrated throughout their genome. They've been evolving separately from any human population for 70 years under artificial selection pressures in laboratories. They're their own thing now. A unique form of life that originated from Henrietta Lacks, but has diverged significantly from any normal human cells. And they're essentially immortal. Barring some catastrophic event that wipes out all the laboratories that maintain them, HeLa cells will likely continue existing long after every person alive today has died. They could outlive our civilization. In some strange sense, Henrietta Lacks might have achieved a form of immortality far beyond what anyone ever imagined. But is that really immortality? Are those cells still Henrietta? Would she have wanted this? Could she have even conceived of it? Chapter 5. Other Immortal Cell Lines. Henrietta wasn't alone. HeLa cells were the first immortal human cell line, but they weren't the last. In the decades since Henrietta's cells were cultured, scientists have developed hundreds of other immortal cell lines from different donors, different tissues, different types of cancer. Each has its own story, its own ethical complications, its own contributions to science. Let me tell you about some of them, because understanding the broader picture helps illuminate what happened with HeLa and what continues to happen today. In 1962, a Chinese woman's lung tissue was used to create a cell line called Chang liver. The researcher claimed these were liver cells from a Chinese individual, hence the name. But later testing revealed they were actually HeLa cells, likely from contamination. The researcher had either been fooled by HeLa's aggressive spread or had intentionally mislabeled them. Either way, the Chang liver cell line was actually Henrietta Lacks' cells under another name. In the 1970s, a man named John Moore was being treated for hairy cell leukemia at UCLA. His doctor noticed that Moore's cells produced unusual proteins that could be valuable for research and pharmaceutical development. The doctor continued taking samples long after Moore's cancer was cured, eventually developing a cell line called Mo. The cell line was patented. It was licensed to pharmaceutical companies. It generated millions of dollars in revenue. John Moore received nothing. He didn't even know his cells were being used commercially until he read about it in a scientific paper years later. Moore sued, arguing that he had property rights to his own cells. The case went all the way to the California Supreme Court, which ruled against him in 1990. The court decided that once biological materials are removed from your body, you don't have ownership rights to them. The doctor and the university own the cell line, the patent, and all the profits. It was a landmark decision that essentially confirmed what had happened to Henrietta Lacks was legal, even if it was ethically questionable. There's a cell line called WI38, developed in the 1960s from the lung tissue of a fetus. This cell line has been used to develop vaccines for rubella, rabies, measles, chickenpox. Billions of people have received vaccines grown in WI38 cells. The tissue came from a single electively aborted fetus in Sweden, donated for research. The family was never identified, never compensated, never acknowledged. There's MRC5, another fetal cell line from 1966, used similarly in vaccine development. The ethical questions around fetal tissue and research are complex and deeply divisive. But what's clear is that a handful of cell donations made decades ago continue to benefit humanity today, while the donors remain anonymous and unrewarded. There's a cell line called MOLT4, derived from a teenage boy with acute lymphoblastic leukemia in the 1970s. His cells are used extensively in immunology research. Nobody knows his real name. The family was never contacted about how his cells would be used. There's the Detroit 562 cell line from a woman with pharynx cancer, SK-MEL from a melanoma patient, HEK293 from embryonic kidney cells. Hundreds of cell lines, each with a story, most with an anonymous donor whose contribution changed science, but who was never acknowledged or compensated. Not all of these stories involve exploitation or ethical lapses. As medical ethics evolved, consent procedures improved. More recent cell lines are typically developed with explicit informed consent, with the donor understanding how their cells might be used. Some donors specifically want their cells to be used for research. They see it as a way to contribute to science, to help others. But the historical cell lines, the ones developed in the 1950s through 1980s, they're complicated. The people who donated those cells, voluntarily or not, couldn't have imagined what would be possible. They couldn't have foreseen that their cells would still be alive and reproducing 70 years later. They couldn't have anticipated the commercial value, the scientific impact, the ethical debates. And in many cases, they weren't asked. Consent wasn't obtained, or it was obtained in a way that didn't fully explain the implications, or it was obtained under circumstances where the person didn't really have the power to refuse. The Henrietta Lacks story has forced the scientific community to reckon with this history. Professional organizations have developed stricter guidelines for obtaining and using human biological samples. Ethicists have written extensively about the rights of tissue donors. Laws have been updated, though they still vary widely between countries. But the cell lines already in existence, developed under old standards, they're still being used. HeLa cells are still the most popular human cell line in research. They're still generating revenue for the companies that culture and sell them. And Henrietta's family still doesn't receive any compensation. There's an argument that says Henrietta is dead, the cells are just cells. They've been so changed by decades in culture that they're barely human anymore. The research they've enabled has saved millions of lives. Isn't that more important than questions of consent and compensation? But there's a counter-argument that says the cells came from a person, that person had family. Taking something without permission, even if you do good things with it, is still wrong. The fact that Henrietta was black, poor, and sick makes it worse, not better. It's part of a long history of exploiting vulnerable populations for medical advancement. Both arguments have merit. Both grapple with real ethical principles. The tension between them hasn't been resolved, and perhaps it can't be. But what's clear is that the conversation needed to happen, and it took 60 years before it really did. Continues with more chapters. Chapter 6. The Ethics of Immortal Cells. Let's talk about the deeper ethical questions that the HeLa story raises, because they're not simple and they don't have easy answers. First, there's the question of consent. In 1951, it was common practice for doctors to take tissue samples from patients for research without explicit consent. The understanding was that the tissue was removed for medical purposes anyway, and any leftover tissue could be used to advance science. Patients benefited from medical advances, so using their tissue for research was seen as mutually beneficial. But this practice disproportionately affected marginalized populations. Teaching hospitals where much research took place primarily served poor patients, Black patients, immigrant patients, patients without resources or education to advocate for themselves. These were the people whose tissue was most likely to be taken without meaningful consent. Henrietta went to Johns Hopkins because it was one of the only hospitals in Baltimore that would treat Black patients for free. She had no choice, and that lack of choice extended to what happened to her cells. Would Henrietta have consented if she'd been asked? We'll never know. But the fact that nobody thought to ask, that her humanity and autonomy weren't considered worth the conversation, that tells us something about how Black patients were viewed in 1951. Second, there's the question of compensation. If Henrietta's cells have generated billions of dollars in commercial value, if companies have profited from selling them, if pharmaceutical companies have used them to develop products they sold, shouldn't her family receive something? The counter-argument is that the cells required enormous amounts of scientific labor to be useful. Researchers had to figure out how to culture them, how to work with them, how to apply them to specific problems. The scientific and commercial value wasn't inherent in the cells themselves, but in what scientists did with them. But that argument doesn't really hold up. Raw materials have value. Oil in the ground isn't useful until it's refined, but we still pay the landowner for the oil. Gold ore isn't valuable until it's processed, but miners still compensate whoever owns the land. Why should biological materials be different? Why should the person whose body produced them have no claim to any value derived from them? The John Moore case established legal precedent that tissue donors don't have property rights to their excised tissue. But legal doesn't mean ethical. Laws can be unjust. And there's a growing movement to change these laws, to recognize that people should have some say in what happens to their biological materials, and some share in any commercial benefits derived from them. Third, there's the question of privacy. Henrietta's medical records were published in scientific papers. Her name was used publicly without her family's permission. Details of her life, her illness, her treatment became public knowledge. Modern medical privacy laws like HIPAA in the United States would prevent this today. But those laws didn't exist in the 1950s, and even now, they're not perfect. Genetic information from HeLa cells could potentially reveal information about Henrietta's living descendants, their risk for certain diseases, their genetic traits, intimate biological details. In 2013, German researchers published the complete HeLa genome online, making it freely available to anyone. The Lacks family objected, arguing that this violated their privacy, revealed information about their own genetics without consent. The NIH eventually brokered an agreement to restrict access, but the damage was done. The genome was already out there, copied, downloaded, distributed. Fourth, there's the question of acknowledgement. For decades, Henrietta was invisible in the story of HeLa cells. Scientists wrote papers using her cells without mentioning her name. Textbooks discussed HeLa as an important research tool without explaining where they came from. The woman was erased from her own legacy. It took a journalist, Rebecca Skloot, spending 10 years researching and writing a book for Henrietta's story to reach the public. Scientists could have done this at any point. They chose not to. Why? Part of it was the culture of science, which often treats biological materials as just materials, divorced from their human source. Part of it was likely racism, the devaluing of Black lives, the assumption that a Black woman's story wasn't important or worth telling. But the result was profound injustice. Henrietta made one of the most significant contributions to medicine in history, and almost nobody knew her name. Fifth, there's the question of what we owe to past donors. The cells exist. They're incredibly valuable for research. Should we stop using them because they were obtained unethically? Would that honor Henrietta, or would it waste the opportunity her cells represent? Most ethicists argue that we should continue using HeLa cells, but we should do so with acknowledgement of the ethical failures that led to their existence. We should tell Henrietta's story. We should ensure that modern tissue collection is done ethically. We should use HeLa cells in ways that Henrietta would likely have approved of, avoiding gratuitously harmful research. And we should arguably provide some compensation or benefit to her descendants. Not because they have a legal claim, but because it's the right thing to do, recognition that Henrietta's contribution was taken without permission and deserves acknowledgement. The Henrietta Lacks Foundation, established with proceeds from the book about her life, provides scholarships and financial assistance to Henrietta's descendants. It's something, but it's a tiny fraction of the economic value that HeLa cells have generated. Is that enough? Is that just... I don't know. But I know the questions matter, and I know that we should keep asking them. Chapter 7. Modern Immortal Cells and Current Research. The science of immortal cells hasn't stopped with HeLa. In fact, it's accelerated dramatically. Let me tell you about some of the cutting-edge research happening right now with immortalized cells. Scientists have developed techniques to deliberately immortalize cells, not just relying on rare cancerous cell lines. They use genetic engineering to insert the telomerase gene into normal cells, giving them the ability to divide indefinitely. These are called immortalized cell lines, and they're created in laboratories for specific research purposes. The advantage of these engineered immortal cells is that they're more controlled than cancer-derived cell lines like HeLa. They have normal chromosomes, predictable behavior, and they're designed for specific purposes. But they also lack some of the resilience and adaptability of natural immortal cells like HeLa. One exciting development is induced pluripotent stem cells, or iPSCs. These are adult cells that have been reprogrammed to become stem cells, capable of turning into any type of cell in the body. And importantly, they can be made to divide indefinitely, essentially making them immortal. The breakthrough with iPSCs, developed by Shinya Yamanaka in 2006, earned him a Nobel Prize. The implications are enormous. You can take a skin cell from a patient, reprogram it into a stem cell, and then turn it into any type of tissue you need. Liver cells, heart cells, neurons, anything. This has huge potential for personalized medicine. Imagine testing drugs on cells that are genetically identical to the patient, predicting how they'll respond to treatment before administering it. Imagine growing replacement organs from a patient's own cells, eliminating rejection issues with transplants. And because iPSCs can divide indefinitely, you can grow as many as you need. You're not limited by the Hayflick limit. In a sense, we've learned to create immortal versions of anyone's cells on demand, but this technology also raises new ethical questions. If we can make anyone's cells immortal, should we? Do people have a right to refuse? What if their cells are particularly valuable for research? Can we create immortal cell lines from people without their knowledge, as long as we don't physically harm them in the collection process? Another frontier is organoids, three-dimensional structures grown from stem cells that mimic real organs. Scientists have created mini brains, mini livers, mini kidneys, all grown from immortal stem cells in petri dishes. These organoids can survive indefinitely with proper care, growing, functioning, responding to stimuli. Brain organoids are particularly fascinating and ethically troubling. They're not conscious, not in any meaningful sense. They don't have experiences, but they have neurons that fire, that form connections, that exhibit electrical activity similar to early-stage human brains. As these organoids become more sophisticated, we have to ask, at what point do they deserve ethical consideration? If we grow a structure that has the complexity of a developing human brain, even if it's in a dish, does it have rights? Can it suffer? Would it be wrong to destroy it? These aren't hypothetical questions. Researchers are wrestling with them right now. And the same principles that we failed to apply to Henrietta Lacks' cells, we need to apply to these new technologies before they become entrenched practices. There's also fascinating research on natural immortal organisms. Did you know there's a jellyfish species, Turritopsis dohrnii, that's biologically immortal? When it reaches old age or is injured, it can revert to its juvenile form and start its life cycle over. Theoretically, it could live forever. Scientists are studying these jellyfish to understand the mechanisms of biological immortality. Could we apply similar principles to human cells or human aging? Could we reverse aging at the cellular level? HeLa cells have actually been used in aging research because they don't age in the traditional sense. By comparing HeLa cells to normal cells that do age, researchers have identified key differences in gene expression, in cellular maintenance, in stress responses. One discovery is that immortal cells like HeLa are much better at repairing DNA damage. They have enhanced DNA repair mechanisms that fix breaks and errors before they accumulate. Normal cells accumulate damage over time, eventually triggering senescence or death. Immortal cells fix the damage and keep going. Another difference is metabolic. HeLa cells have altered metabolism, processing nutrients and energy differently than normal cells. This gives them advantages in survival and growth, but it also makes them behave abnormally, which is part of why they're cancerous. Understanding these differences might eventually lead to treatments that could extend human lifespan, or at least health span, the number of years we live in good health. If we could enhance DNA repair mechanisms without triggering cancer, we might slow aging. If we could modify metabolism to be more efficient without becoming pathological, we might extend cellular lifespan. But here's the paradox. The very features that make cells immortal, that allow them to escape the Hayflick limit and divide indefinitely, are the same features that define cancer. Immortality and cancer are closely linked. You can't easily have one without the other. This is why cancer is so hard to cure. We're not just fighting a disease, we're fighting cells that have achieved a form of biological immortality, cells that have escaped the safety mechanisms that normally prevent runaway growth. When you think about it, Henrietta's cancer was extraordinary in a terrible way. It grew so fast, was so aggressive, that it killed her in 8 months. But those same properties, that aggressiveness, that refusal to stop dividing, made her cells invaluable for research. Her death and her immortality are two sides of the same biological coin. Chapter 8. The legacy, what Henrietta means today. So what is Henrietta Lacks' legacy? What should we take away from her story? First and most obviously, her cells have saved countless lives. The polio vaccine alone prevented millions of cases of paralysis and death. The cancer treatments, the HIV therapies, the countless drugs tested on HeLa cells, all have contributed to human welfare in measurable, significant ways. Every person alive today who was vaccinated against polio owes something to Henrietta Lacks. Every person who survived cancer thanks to chemotherapy protocols tested on HeLa cells owes something to Henrietta. Every person who's living with HIV because of effective treatments owes something to Henrietta. But acknowledging that contribution requires acknowledging the woman herself. Not just the cells, but the person they came from. A woman who loved her children, who worked hard, who died young, who never knew that part of her would live on. Second, Henrietta's story is a stark reminder of medical racism and exploitation. The history of medicine is full of cases where Black bodies were used for research without consent. The Tuskegee syphilis experiments, where Black men were deliberately left untreated to study the disease's progression. J. Marion Sims, the father of gynecology, who performed experimental surgeries on enslaved Black women without anesthesia. Henrietta Lacks is part of this history, and understanding her story requires understanding this context. Medical research has built much of its knowledge base by exploiting vulnerable populations. The poor, the imprisoned, the colonized. People who couldn't refuse, who had no power, who were seen as less than fully human. We've made progress. Medical ethics has evolved. Institutional review boards, informed consent requirements, protections for human subjects, these are all real improvements. But they came too late for Henrietta and countless others. And we need to remember that progress came because people fought for it, because victims and their families demanded change, because journalists and ethicists forced the medical establishment to confront its failures. Third, Henrietta's story raises ongoing questions about ownership of biological materials. Your DNA is unique to you. It reveals intimate information about you and your relatives. If scientists can derive valuable insights or products from your genetic material, do you have a right to control that, to benefit from it? Current laws say mostly no. Once biological materials leave your body, you generally lose control over them. But there's increasing recognition that this is unjust, that we need better frameworks for respecting bodily autonomy and ensuring fair compensation. Some researchers advocate for a model where tissue donors are treated as partners in research, giving informed consent for specific uses, receiving credit in publications, potentially sharing in any commercial benefits. This would be more work for researchers, more complicated than the current system, but it would also be more ethical. Fourth, Henrietta's story highlights the importance of diversity in medical research. For decades, medical studies were conducted primarily on white men. Treatments were tested on white men. Dosages were calculated based on white men's bodies. And then those treatments were applied to everyone. Despite differences in how medications affect people of different sexes, ages, and genetic backgrounds. HeLa cells, ironically, became a tool for diversifying research. They're cells from a Black woman, and they've been used to develop treatments that benefit all populations, but we still have a long way to go in ensuring medical research includes diverse populations, tests treatments on people who will actually use them, and doesn't assume white male bodies as the default. Fifth, Henrietta's story is about recognition, about the human tendency to reduce people to their utility, to forget the person behind the tool. Scientists used HeLa cells for decades without thinking about Henrietta Lacks as a person. She was just a label, a convenient designation. But Henrietta was a person. She had dreams and fears. She loved her children. She suffered, she died, and she made a contribution to humanity that deserves recognition. There's a memorial to Henrietta at Johns Hopkins now, installed in 2018, nearly 70 years after her death. There's a statue of her at Royal Fort House in Bristol, UK, where the first HeLa cells arrived in Europe. The World Health Organization honored her in 2021. She's been the subject of books, movies, documentaries, academic papers. Finally, after all these years, Henrietta Lacks is getting the recognition she deserved all along. But recognition isn't enough. The systemic issues that allowed her exploitation still exist in various forms: healthcare inequality, medical racism, the commodification of human biological materials without consent or compensation. These problems persist, and solving them requires ongoing effort and vigilance. Henrietta's great-granddaughter, Jerry Lacks Y, has become a science advocate, speaking about her great-grandmother's legacy and pushing for better ethical standards in research. She represents the fifth generation of the Lacks family, carrying Henrietta's story forward. And in a very real sense, Henrietta herself lives on. Not just in memory, not just in the impact of HeLa cells, but literally. Part of her is still alive, still reproducing, still contributing to science. 70 years after her death, cells with her DNA are alive in laboratories on every continent. That's a strange kind of immortality, not the one anyone would choose, but remarkable nonetheless. Chapter 9, Conclusion. The Woman Who Wouldn't Die. Let me end with a thought experiment. Imagine if Henrietta had known what would happen. Imagine if, in 1951, a doctor had explained to her that her cancer cells were unusual, that they could potentially be cultured indefinitely, that they might contribute to medical research for decades to come. Imagine if that doctor had asked, "Mrs. Lacks, may we use your cells? They could help develop vaccines, understand diseases, save lives. But we can't predict exactly how they'll be used or where they'll end up. Do you consent?" What would Henrietta have said? Her family has said they believe she would have agreed. Henrietta was a giving person, devoted to her children, her community. If she'd understood that her cells could help save other mothers' children from polio, other families from cancer, she likely would have consented. But we'll never know for sure because nobody asked. And that's the tragedy at the heart of this story. Not that Henrietta's cells were used for research, not that they contributed to medical advances. Those are good things. The tragedy is that Henrietta's autonomy, her humanity, her right to make that choice were never respected. She was treated as a source of biological material, not as a person, and that treatment was shaped by racism, by poverty, by the power dynamics between doctors and patients, between white institutions and Black individuals. The HeLa story is simultaneously a story of scientific triumph and ethical failure. Both things are true. Both things matter. And the reason Henrietta's story resonates so powerfully is that it makes us confront uncomfortable truths about how medical progress has been achieved, about who pays the costs, about whose contributions are remembered and whose are forgotten. Every time a scientist uses HeLa cells, they're using part of Henrietta Lacks. They're benefiting from her involuntary contribution, and increasingly, thanks to the work of her family, thanks to journalists and ethicists and advocates, they're required to acknowledge that. There's now a movement in science to always cite Henrietta Lacks in papers that use HeLa cells, to include her name in acknowledgments, to treat HeLa not as an abstract cell line, but as human tissue from a specific person who deserves credit. Some journals have made this a requirement. Some institutions teach students about Henrietta as part of their ethics curriculum. Slowly, the scientific community is changing how it thinks about and treats HeLa cells. But beyond HeLa specifically, Henrietta's story has sparked broader changes in how science approaches human tissue research. Consent forms are more detailed. Ethical review is more stringent. There's greater awareness of historical injustices and efforts not to repeat them. None of this happened automatically. It happened because people demanded it. Henrietta's family, journalists, ethicists, they forced these conversations. They made it impossible to ignore the human cost of scientific progress. And that work continues, because new technologies keep creating new ethical challenges. Genetic engineering, stem cells, organoids, artificial intelligence in medicine, all of these raise questions about consent, privacy, ownership, equity. If we learn from Henrietta's story, we can handle these new challenges better. We can build systems that respect human dignity while advancing science. We can ensure that the benefits of medical research are shared fairly, that the costs aren't borne disproportionately by vulnerable populations. Henrietta Lacks died on October 4, 1951. She was 31 years old. Her cells are still alive, still reproducing, still contributing to scientific discoveries. In a very literal sense, part of Henrietta is immortal. But what matters most isn't the biological immortality. It's the recognition, finally, that Henrietta Lacks was a person, a woman with a name, a life, a family, a story worth telling. She wasn't just HeLa, she was Henrietta, and now the world knows her name.